Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. My name is Ayesha, Senior Life Science Journalist at Xtalks.com, and this week I am delighted to introduce another special guest on the show. With us we have Dr. Craig Erickson, who is the Chief Medical Advisor at Spinogenix, which is a company innovating treatments for neurological disorders such as ALS, Alzheimer's disease, Fragile X syndrome, and among others. Dr. Erickson has been conducting translational research in neurodevelopmental disorders for 20 years. He is the leader of a large translational medicine program at Cincinnati Children's Hospital Medical Center, built to enhance the ability to detect brain target engagement with treatment on an individual patient level. This has enabled Dr. Erickson to, from the earliest stages of human drug trials, detect potential meaningful functional changes in human subjects while additionally describing which specific patients may best respond to treatment. Dr. Erickson holds several investigator-initiated INDs and is the inventor of several patents describing new treatment approaches in his fields of study. His work has been extensively supported by the NIH, the CDC as well, and the U.S. Department of Defense, as well as stakeholder foundations and biotechnology companies from around the world. Dr. Erickson is a professor of psychiatry at the University of Cincinnati College of Medicine affiliated and the director of the Cincinnati Fragile X Research and Treatment Center. To learn more about the work that Dr. Erickson is helping lead at Spinogenix, particularly in the area of Fragile X, let's turn to him now. Dr. Erickson, thank you so much for being on the show with us today.
Well, thank you for having me. Really looking forward to it.
Likewise. And I'd like to start off by asking you, you've been in this space for over 20 years, and so I'd love to learn a bit more about your journey and what ultimately drew you to Spinogenix, and how your role as Chief Medical Advisor helps to shape the company's research and clinical development strategy.
Yeah, no, I went into medicine in medical school decades ago, really to work with people with developmental disorders and who had significant neurocognitive deficits, speech, other aspects of communication, learning, and behavioral deficits. And I actually, first met people with these disorders working at summer camps when I was in high school and college up until medical school. And I really experienced at that time firsthand a lot of the impairments and challenges that individuals had throughout the age range, really requiring lifelong care or lifelong adult supervision forever. And just knew the extent also of interfering behaviors and anxiety and agitation and related, often driven by a lot of the communication and sensory deficits. So that was my original driving force. And then as I moved into medicine and new treatment development and conducting clinical trials early in my career, in autism, we got a couple of medications approved, one aripiprazole Abilify and another risperidone Risperdal to treat aggression, self-injury, and severe tantrums in autistic kids. And then I was very optimistic following those trials, thought, boy, we'll have a lot of other success treating core cognitive and communication and sensory and anxiety impairments. And those did not come. And we had a lot of failed trials subsequently. In fact, my career is littered by, 20 years of not meeting primary endpoints in clinical trials. And reflecting upon that has really led me to the journey with Spinogenix. And it's that we want to match individuals to specific novel new medications that may provide benefit to them clinically, but at the same time, we want to evaluate those treatments in highly quantitative ways. Because what I've learned for years is, as a clinician, as a clinical scientist, and families, we all want our patients to get better. So if we rely on checklists and scales of, this person's doing better, they're communicating more, they're calmer, they're more attentive, we get huge placebo effects. So I've really had a shared vision with Spinogenix in everything they do, whether it's their work in ALS or Alzheimer's or specifically in Fragile X that we're talking about today. When they do their first trials, they evaluate classic outcome measures. How are folks doing clinically? What are doctors and others observing? What are families observing? But they also do what we call neurophysiologic testing or work with EEG and work with something called transcranial magnetic stimulation to look at what are the treatments doing to brain activity. So that's where my link is with Pete Vanderklish, who's the chief science officer at Spinogenix. We used to have collaborative Fragile X research years ago. And I knew that he was involved with this group and that they were dedicated to looking at things that way, asking, what quantitatively, what's actually happening with brain function. And that's what we did in the first Spinogenix Fragile X trial. And I think it's been quite successful for that reason. So it's really out of years of failure to retool our clinical trial approach. And a lot of biotechnology companies don't have that approach. They still have an approach of just giving drugs to large groups of individuals and seeing how things shake out. But we've had so many failed late phase trials in Fragile X and autism and related disorders that I think we need to do it differently. And I share that vision with Spinogenix.
Thank you so much for sharing that journey and really those insights into what ultimately brought you to Spinogenix and the type of work that you're leading there. And for some of our listeners, our listeners are industry based, but for those who may not be so familiar with Fragile X, could you tell us a bit more about the disease and why it has such high unmet need and why it's been so difficult to target it therapeutically?
Fragile X is the most common single gene cause of autism, and it's the most common inherited form of developmental disability worldwide. On average, it affects about 1 in 4,000 individuals. It's an X-linked genetic disorder, and all males have one X and one Y, and females have two X's. So with only one X, every male with Fragile X is affected. They have a repeat expansion, a CGG repeat expansion in their Fragile X gene that results in gene methylation and then reduced or absent protein, Fragile X protein expression. Females, being XX, are really a flip of a coin whether they're affected or not. So 50% of females with Fragile X have significant clinical impairment. 50% don't, but impairment is uniform in males. So that's where we took one important step with the initial Spinogenix work. We focused it in on the classic males that are not making the protein. But, the presentation of Fragile X, it's marked by moderate to severe intellectual or developmental disability, both expressive and receptive. So what you say and what you understand, communication deficits, significant sensory processing issues. So individuals that have difficulty understanding certain sounds and taking in both visual and auditory information, ton of anxiety or being on edge. And all of these impairments are lifelong, so they don't go away. And we have a very large Fragile X center here at Cincinnati Children's Hospital where I am. We see patients of all ages, almost 1,000 individuals with Fragile X. And the individuals, by and large, in particular, all of the males, require lifelong support, supervision, and care. And they're not able to care for their daily needs like transportation, preparing food, even some individuals with self-care and toileting. So it's a significant impairment. It's extremely costly. And it just really weighs on the individuals and families. And as far as, why don't we have approved treatments? We've tried a lot of targeted treatments. We've done a lot of preclinical animal work, and I have an animal lab myself too, where we thought we were, onto something or had some improvements, but it didn't translate into human work. And that's where the EEG that I talked about before, studying brain electrical activity with EEG nets on the head, we've leveraged a decade plus of NIH funding to establish the same EEG markers in mice that have the gene knocked out as in humans with Fragile X. We see the same electrical activity abnormalities. So now when we do first in human Fragile X drug trials, we can really target those electrical brain signals and see if they're correcting or not. And it often doesn't take a lot of patience. It's either working or it's not. So that's how we could get by with our first Spinogenix trial with just 10 adult men with Fragile X, because we're really focusing on, is the brain's own electrical signaling, is it normalizing or not? And that comes out of, again, years of research. And we think this more targeted approach in Fragile X, with males that have the specific EEG findings for initial studies, really de-risks what we're doing versus combining males and females and all ages at once, where there's a ton of variability.
Yeah, wonderful. I think, one of my next questions was going to be about really what are the most significant endpoints in trials, and so EEG being a powerful one as a powerful readout. And would this be a powerful readout throughout the patient journey, whether we're talking about to assist in diagnosis through to monitoring treatments and looking at outcomes as well?
I think it's a really great measure for monitoring with treatments and target engagement or having an effective, brain change. I also think there's developmental benefit to it. We're on the cusp of doing another big NIH research project to look over time in children and adolescents to see when some of these patterns come on. We have some hypotheses, for example, that it's during adolescence itself with the onset of puberty. So if we can sort of pinpoint the timeline of when some of these patterns happen, I think when we get into earlier interventions, earlier treatment, potentially, early medication studies in younger children, we can block the abnormalities for even developing. So I think that's kind of, the end game. Other measures that are really important, we've talked some about EEG, but Spinogenix has employed computer-based cognitive testing using a modality called the NIH Toolbox, that I've been part of a national consortium in the United States that's developed it for use in Fragile X and related disorders. And that's performance-based. So it's really like, can an individual follow a pattern? Can they do matching? What's their memory like? It's very tangible versus, again, having placebo response risks.
Mm-hmm. That's wonderful to hear that. Again, a combination really of different biomarkers, assessment tools is really helping to drive trials forward and to generate robust data, which is of the essence. And in terms of running clinical trials, and as you mentioned, success rates and all of that is always a challenge. What are some of the key challenges in terms of, particularly running trials in rare neurological diseases like Fragile X, and especially in pediatric populations?
Yeah, I think you really need to be engaged with the stakeholder community. We do research, but we provide clinical care for anyone across the United States and the world that wants it. So we are not sort of isolated in our pursuits. And I think we're about building relationships and having, a lot of my patients I've seen for 20 years, 15 years, 12 years, 10 years. I just saw a family from Northern Ohio and probably seen their son for 13 years, and he's a young adult now. And so it's about relationship building and engagement, because it is a very rare disease. And I think that's daunting from new treatment development. How do you find the individuals? Well, we have one of the largest Fragile X centers in the world here in Cincinnati, Ohio, in the US. And I think in part of that is because we're as committed to clinical care as we are to cutting edge research. So we have a number of physicians and psychologists and nursing staff and other care providers that we are helping folks at all levels. Social work engagement, helping them with services, interfacing with the school, those kind of things. It's not just the research picture. I think that's how we overcome a lot of the engagement. And when you offer that, people have options if they want to participate in research. And I think by and large, we find they do. We get upwards of 90% of families participating in some form of research, but it's not required. It's not just a research-only clinic. And I think that's different. There's a few places like this in the United States that are very high volume, but I think it's because we're also providing care And you're not just coming for a trial, right? You're in it for the long haul. If a trial fails and goes away, or if it's not a fit and someone doesn't participate, you're still part of the family at our Fragile X Center. I think that's extremely important.
Mm-hmm. That sounds wonderful. Like it's really a space where patients can feel that they're getting, again, like treatments, and then also being a part of trials that can lead to new and improved treatments. And so I think that's very powerful to have that whole infrastructure in place at a place like Cincinnati Medical Center. It's Cincinnati Children's Hospital, is it? Yeah, Cincinnati
Children's Hospital. It's patients of all ages, so it's a misnomer. So
okay.
We're at a children's hospital, but
Right.
in a lot of ways, folks with significant developmental delay, they don't have great adult resources. So, my oldest patient's over 70, and our youngest patient maybe 5 weeks of age. So
Wow.
it spans all ages, but we're anchored at Cincinnati Children's Hospital. Yeah.
Yeah. No, that's wonderful. Again, that infrastructure, that network is so important. And when we talk about different stakeholders, of course, the patient and their caregivers are such an important piece, the central piece, if I may say, of course, in any kind of research and any kind of really supportive care, and really building on that. But who are some of the other stakeholders, in your opinion, who are important to have at the table, whether we're talking about industry partners, academia, and again, how do you bring all of those stakeholders together, given their potentially differing interests? But I would imagine in this day and age, of course, that the patient is central to a lot of these stakeholders.
Yeah, I think the families, the parents, other caregivers, the patients, other relatives are central, but we do have to interface with a lot of other systems, right? And so for us, we work a lot with insurers, school systems, state level governments. To a degree, we're advocating with the federal government a lot. The National Fragile X Foundation partners with us and other centers on yearly advocacy days. So, the Department of Defense in the United States specifically has Fragile X as a line item for research funding because of that advocacy. I've been lucky enough to have what are called Fragile X NIH Center grants that are driven by family advocacy with the U.S. Congress. And I think we've been able to lever a lot of our federal research dollars to develop techniques and understanding about brain development, the ability to use these tools like EEG and the computer testing in trial settings. But I often tell people, the NIH is not going to get new treatments approved. That's really not their role in Fragile X, and we have to partner with industry and biotechnology groups and those that are seeking to innovate in the field to move things forward. So I think we need to be levering in multiple directions at the same time, right? Like all the techniques that we're utilizing with Spinogenix, they were supported by a lot of our federal NIH work to develop these EEG techniques, develop the computer testing, do pilot studies with various available drugs supported by the NIH. But they're not going to be moving treatments at the NIH into phase 2B and phase 3 large clinical trials to approval. So that's where we need to take those lessons learned off and tell people that's how we're levering taxpayer dollars to learn really important information and then broaden out how applicable it is by finding other partnerships. And that's exactly what we're doing with Spinogenix. And without both in all of these different angles, we couldn't develop novel treatments this way.
Indeed. And when we talk about different partners and different stakeholders, also different aspects of drug development, we talk about the regulatory end of things as well. And so what is the current landscape regulatory-wise in terms of developing treatments and submissions for approval, whether it's with the FDA? And so what is the current landscape like for rare diseases like Fragile X? And I know that we've come a long way in terms of the FDA having various pathways, accelerated approvals, and of course the orphan drug designation's been around for decades. And so in your opinion, what is the current landscape and how has it evolved over, let's say, the last several years or so?
Well, I think it's involved in a landmark way that the FDA is open to the concept of performance-based outcome measures and not sole reliance on clinician and caregiver ratings, where again we have challenges with significant placebo effects. So, you know, Fragile X sits in the psychiatry branch of the FDA's evaluation group, and a nice thing is they've had some stability there with the folks that have been handling Fragile X discussions. I think that's helpful. But, we've been able to consistently advocate with the FDA. So I've participated with the National Fragile X Foundation and others with various stakeholder information sharing sessions with the FDA to talk about what's meaningful to families and caregivers and others directly impacted by Fragile X. And I think that's led to this concept of being able to utilize, for example, potentially computer-based testing as a primary outcome to look at how someone is functionally changing with treatment. I think that is a breakthrough versus reliance on checklists solely. Because the challenge with Fragile X is it's a developmental disability. The vast majority of the patients cannot necessarily accurately or clearly communicate the state of their being. So that's where performance, what you can do in a test that relates directly to your IQ, relates directly to your adaptive behavior, or what you can functionally do, can be a really reliable outcome in a potentially approvable endpoint. So that toolbox is a reliable outcome we've been able to employ in our Spinogenix first in Fragile X trial. And the FDA is open to it being approvable. And 10 years ago, there was no discussion like that. Everything was sort of pen and paper, checklist based for outcomes.
Wow. That's very encouraging and very promising to hear how the space has evolved, and especially on the regulatory end. And so actually, I'd like to ask you more about Spinogenix's approach to Fragile X. And so I know that the company is focused on restoring synaptic connections. And so how does that approach differ potentially from past approaches for Fragile X and also currently in the field?
Yeah, so, I think last summer I announced on behalf of Spinogenix, their initial work in Fragile X focused on large conductance potassium channel modulation. So this is a neurotransmitter system that's repeatedly been shown to have abnormal function in Fragile X preclinical models. And Spinogenix was able to take one of these large conductance potassium channel modulators, or BK channel modulators, that already had a lot of human safety data and bring it quickly into Fragile X syndrome. So we could dose 10 men, 18 to 45 years of age, with 800 milligrams of the SPG 601 drug or placebo, and do that study, late summer, fall in a few months, and get some data very quickly. And that's because of the existing safety profile of the drug. But, Spinogenix is the first group that's evaluated this mechanism in Fragile X. And again, we were able to do it, quite quickly because of the established profile of the drug safety-wise. It didn't take years of early phase 1 trials, because that work had already been done. We could just get it right into Fragile X and use all the markers we talked about to see if we're having a positive brain effect or not.
Mm-hmm. Wonderful. And so how do you assess the current state of drug development for Fragile X, of course, with the very innovative approaches being applied at Spinogenix? But overall, how have you seen the field evolve in the neuropsychiatry space? Number one, I guess, in general, is there more focus on, I think as a whole, in general, I think we can talk about it being kicked off really in oncology with precision medicine, targeted therapies. And so how has that now arrived in other fields like neuropsychiatry, and how are the approaches really differing today as opposed to the past, and where do you see the field and space heading in the next couple of years?
Yeah, I totally agree. I think oncology is the greatest example. In fact, when I give my personalized medicine and neurodevelopmental grand rounds talks, I refer to example, breast cancer, BRCA testing, and aligning people with specific, therapeutic courses. So, for Fragile X, there's one simple adjustment. It's the idea, as I talked about at the beginning, that males and females have different levels of impairment. And there's also something called mosaic males who are a mix of kind of being Fragile X carriers and the full mutation. And my lab started testing Fragile X protein in blood almost 10 years ago and found that it's really much more of a continuous deficit than an absolute deficit as a turn on and off, that maybe a third of males with Fragile X actually make zero of the Fragile X protein, and then two-thirds are making low levels. But that's one example of biologic variation that we have then brought forth and evaluated and phenotyped or looked at the human degree of impairment in behavior and found clear relationships. So now it's really in neuropsychiatry broadly and in Fragile X, and I do a lot of work in autism and other developmental disorders. It's about what individuals are best responding to treatment. How can we parse even a single gene disorder and understand who the best responders may be? It may be that males making zero or trace amounts of protein are having a very robust clinical effect and a milder effect in females and mosaic males. And that's fine, but we have to approach it that way ahead of time and not just throwing everybody in. We do the same thing in autism right now, where we're trying to focus less on autism clinical trials for everybody with autism. And we are instead focusing on folks with specific, behavioral patterns, biologic findings, co-occurring symptoms. Like we've done some work in the gastrointestinal sphere and in focusing in on those subgroups. So I think that's the way to go. And Spinogenix embraces that. And it's larger than neurodevelopment, but I think even in other work Spinogenix is doing in serious brain disorders, it's about, when we enter that field, what is the treatment doing to brain function? And how are we describing that functional change in individuals and not viewing everyone as exactly the same? I think that's just the mindset that's essential for a road to success.
And in Fragile X, are there similar challenges in terms of, let's say, observing at the molecular level or through a biomarker readout where you may elicit a change in a certain biomarker with the drug, but maybe the clinical benefit may not be as robust, as we've kind of seen those kinds of challenges in Alzheimer's, let's say, for some of the newer approved drugs. And so is that something that, how do you balance things of that nature?
Yeah, well, I think, we balance it in a number of ways. And I've been doing biomarker work for about that same 20 years and all sorts of ways of measuring things. I've done, work with blood cellular signaling cascades and looking for changes. And what I've really come upon is high density EEG, or this EEG with real dense data from the brain, has been the most clearly related to clinical impairment in severity of any markers that we've used. And then the key, the real secret sauce, is not using it alone, but pairing it with that computer-based, potentially FDA approvable endpoint testing with the NIH toolbox. So we strongly relate the brain electrical activity change to the functional change with the toolbox, but we're not asking for EEG as an approvable endpoint. We're using it to show target engagement, proof of principle, really aid that go, no-go decision, because we've published a lot in the last 10 years on medicines and Fragile X that do absolutely nothing to EEG signal. And in doing all of that, we've shown that the signals are very consistent within individuals, which is important. There's not a lot of random fluctuation. And then if we're able to rescue or improve the signals, at the same time, if we can look at what someone's able to do from a performance standpoint, what they can functionally do, can they engage a test? Can they recognize a pattern? What's their memory like? Can they inhibit a response? Can they change sort of sets in their thinking at the same time? That's bringing it together, right? We're not just, because you could say who cares if this EEG signal totally normalizes? Well, if we relate it strongly to the phenotype, if we show it's a reliable measure, and then at the same time, in that patient whose EEG is normalizing, we show that their ability to recognize patterns, their memory improves, and that's a potentially approvable endpoint. That's really saying something for those go, no go decisions into larger studies. I think that's the progress we've really made over the last decade.
Wonderful. And so, looking ahead, Dr. Erickson, what milestones are you looking forward to in the coming year and in the upcoming couple of years from Spinogenix, particularly, of course, in Fragile X?
Yeah, I mean, I think in Fragile X, we're really looking to, upscale the work into that classic large, multi-site, double-blind, placebo-controlled, trial, which it is an orphan disorder. So, the hope is only one potential registration trial is necessary. And the work's being done now to really design that future endeavor and take that first target engagement data that we had to make those decisions, de-risk the whole project, and move that forward. So, I think on the time frame you describe, it's moving toward a true registration level, trial in Fragile X, which we're super excited about. But I think those decisions are going to be made on this early work that's highly quantitative, that's going to aid subject selection, aid outcome measure determination, and then really de-risk what you're doing. Because we can't keep having all of these, quote, failed phase 3 trials that don't hit primary endpoints. It becomes a rinse and repeat cycle. And that cycle is driven by early Fragile X study that's not taking these very quantitative, functional approaches to what is a drug actually doing to brain activity, right? We sort of will ourselves towards another large Fragile X study with the best of intentions when maybe we should not be going in that direction. But Spinogenix is not taking that approach, and that's why we're able to partner so successfully.
Mm-hmm. And in looking back on your journey and coming up to Spinogenix, what would you say, in your opinion, as you mentioned that, you've been part of many failed trials, which is the norm in drug development. It's like something like an 80% failure rate. And so how do you keep going? And what are the lessons really from those setbacks, the crucial lessons that you've learned, and what would you say to aspiring entrepreneurs and clinicians who, like yourself, may be wanting to dive into that intersection between really treating patients in the clinic and then also doing the research, translating that to the clinic, and vice versa. So what advice do you have for upcoming drug developers?
Well, I think, you have to start with that, hypothesis. Why is this applicable in a certain condition like Fragile X? Gather translationally relevant preclinical data, if at all possible. We've been doing a lot of translational electrophysiology. We do multi-electrode array EEG in the Fragile X knockout mice. We can do that in other models of neurodevelopmental disorders to see in a mammal model where you're moving signal. And then I can tell you, for example, in Fragile X, okay, the signal change you're seeing in a mouse model, that is consistent with rescue of this EEG abnormality in a subgroup of individuals with Fragile X. And you can hone right into that group in your first in disease study. And you can bring EEG and the computer-based testing into that. So you can greatly lower your costs entering the rare disease space. We did the Spinogenix study with 10. I mean, we're talking 10 to 20 patients, potentially single or brief chronic dosing to make these evaluations. And we process our EEG data the same with the mouse multi-electrode array that we do with the human 128 lead EEG. So we really have a tight synchronized translational pipeline. And again, it lowers the entry effort to get into it, and it allows folks to make some pretty quick decisions on if we're seeing target engagement or not. And again, we're not worrying about placebo response in that way in the initial Fragile X small, phase 1B, 2A trial. So I think that's a really de-risked approach that resonates with treatment developers of, hey, we can get into this very well phenotyped, rare orphan disorder. They have some really strong translational markers, so we can do some quick preclinical work, rapidly extrapolate into what we may see in humans, and then we can recapitulate that way of evaluating brain activity with the EEG nets in our first in human study. And then that sets us up to quickly vault, or leave and evaluate futility quickly too. So I think that's a methodology that's really resonated. And we're doing the same thing with subgroups of individuals with autism that we're doing with Fragile X and other neurodevelopmental disorders. But I think that is, as a clinician scientist, just the way I think we have to do it, because we failed in the past. In the old ways, we're just involving hundreds and hundreds of patients with maybe not the strongest hypotheses, maybe sometimes strong hypotheses, but challenges with outcome measurement. And we've overcome that.
That is so wonderful to hear. And Dr. Erickson, thank you so much for helping overcome a lot of those challenges with the work that you've led in this space and that you continue to lead at Spinogenix. Thank you so much for it. And thank you so much for your insights and for sharing some of that work that you're helping lead in the space. And we thank you so much for your time today with us.
Absolutely. Thank you.
That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next time. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.